us understanding like some disease and how it works and like reveal therapeutic target. So I split my presentation in like two parts. The first part would be like mainly science. So basically what we're doing in the lab. And after, like as the organizer said, I'm gonna present a little bit more about like how, like my journey as a scientist. So where I come from, what I've done, and how I end up working in Toronto, because I guess that with my accent, you guess that I'm not like Anglophone, I'm from France, and just like going through all the journey. So to begin, I like to like give some like really big image of the problem. And I think that when you look at the human body, so human organism, it's really interesting to look at these numbers, because human organism, it's like a really complex machinery. So if you take like a whole human body, when you look at the number of cells in human, it's like gigantic, right? And that's the first level of complexity, but you have like several levels of complexity. So all these cells, for example, are distributed between around like 200 cell type. In each cell, you have like 23 pair of chromosome. You have like 20,000 to 20,000 to 25,000 gene, and you have like 3 trillion base of DNA. So when you look at all this number, you see that it's kind of like a miracle that the body worked as it worked, and that the state where the body is like maintained by the complexity of all these variables working together. So like you can think about the body as kind of a balance, right? So when you're like on good health condition, no disease, everything is balanced. So if you have like more cell proliferation, you need more cell death to maintain number of cell. You need to like regulate all these variables. And that's like defined as the tissue homeostasis. So tissue homeostasis is basically like the state where the body is balanced and where everything like go well, right? But the problem is that on a balance, when you have like an increase in one variable, so let's say like cell proliferation, for example, like this minute dysregulation can lead to the disease progression. So there is several examples of disease progression. I'm pretty sure that the most common that you're thinking about is cancer, because it's basically an increase in cell proliferation. And just this minute dysregulation lead to disease development. So I'm gonna focus a little bit more on cancer because it's what we're like working on mainly on the lab. But if you have like at the end other question about like other diseases, do not hesitate at all. So when you think about cancer, you think about like, okay, we have a tumor, so it's like a lot of cancer cells grouped together that form this tumor. But cancer is actually like much more complex, because like in the 90s or in the 80s, it was the case. We were thinking about like, oh, a tumor is like a big mass of tumor cell. But like as the science like understand how it works, we understood that the cancer, and especially what we call the microenvironment, so all like all the features that are around this tumor are really important. So you have all these tumor cells, but in like the sides of the tumor, we have like fibroblasts that are stromal cells. So you have like mainly two types of fibroblasts, the normal one that are like here in green, and the CAF, so cancer-associated fibroblasts that are in pink. So that's kind of like fibroblasts that are like modified by the cancer cell to help them. So they're playing the role of cancer cell. And on top of that, you have like extracellular matrix, because like your tumor grow in the tissue. So you have all this collagen, all this protein of matrix that help to maintain the organization. And all these features have a role in tumor progression. And again, another level of complexity, you have like immune cells. So let's say like macrophage, dendritic cell, T cells, B cell, and all these cells are attracted here by the inflammation caused by cancer. And also all these cells have a role in cancer progression. And there is like several studies that show that, for example, the number of immune cells in the tumor is associated with the prognosis and with the chance of survival for patients. So all these parameters are really crucial when you speak about like cancer development. And again, on top of that, you have like the last, I would say like the last level of complexity, which is that the tumor, when you look at the tumor, it's like on a specific location. And this location, there is like blood vessel around. So the tumor is not like by itself, right? It's on the whole organism. And what is in the blood circulation, like basically what feed the cancer cell is what comes from the blood circulation. So the position of the cell compared to the blood vessel will determine how much like basically food they have, how much oxygen they have. So there is like gradient of all these molecules between the middle of the tumor. You can see my mouse or not?
Yes, we can.
Oh, okay, perfect. So when you're like on the middle of tumor, you have less oxygen, you have less nutrient, but you have more waste because like the waste also are like expulsed by the blood circulation. So right, you have like all these gradients, and this full like picture, it's what we call like the cancer microenvironment. And now it's like a really, really big field in cancer research. So what you have to just remember is that a tumor is not only cancer cell, it's like a full network of different cell type, different like chemical cue, physical cue, and all of that like form this network, and all this interaction regulate each other. And that's basically like one of the biggest questions on research now, is to understand the full network, right? So how to understand like how that works basically. So right now scientists use mainly like two main models. So they use in vitro model. So it's basically like cultivating a cell in 2D in plastic dish. And on the other side, they use in vivo model. So mouse mainly for cancer, and some like rat model, but it's mainly mouse. So both this model have like advantage and disadvantage, like all the models. So if you look a little bit more in detail about this advantage, disadvantage, in vitro model are really cheap because it's basically like just a box of plastic. They are like super easy to use because you just put your cell there, the cells spread and they grow, and they are high throughput because as it's cheap and as it's like simple, that make it high throughput. So you can like test like thousands of drugs, for example, on your cell in 2D. It's really feasible. There is like robots and all that kind of stuff that increase the throughput. The problem with in vitro model is that it's really often too simple because, as I mentioned before, like there is not only one type of cancer cell, and all the fact that there is no architecture, no matrix, not multiple cell type, mean that you kind of losing the power and like the interest in having this full model. So on the other side, you have like the in vivo model that are like more like close to the reality because you're in a full animal. So you have like all this architecture and all the other type of cell. Even if you're in a mouse here, so it's kind of like a little bit different than human, but it's more easy like to translate a result from mouse to human than from in vitro to human. But the problem with in vivo model is that they're really, really expensive. They are super low throughput. So let's say that you want to try like 1,000 compound, you count in mouse, right? Because you need like 10 mouse per group plus control. So you end up having like in your animal like 2,000 or 3,000 mouse, and that's not like feasible, right? And also there is like big ethic concern about like the use of animals model, like to decrease the number of animals is kind of like now a priority in research. So with these two models, you see that basically there is no like one perfect model. And on the middle of these two models, there is like an intermediate model that is more recently developed, that is not having like all the advantage of both model or all the disadvantage, but it's kind of in between. So like, it's not as easy in vitro, but it's like much easier than in vivo. It's the same for the reality because you can have like this 3D structure, you can make like co-culture. So you're adding more complexity in tissue engineering model. It's cheap. I mean, it's not as cheap as the 2D, but it's definitely like much cheaper than in vivo model. And the throughput, depending on model, can be like from low to like medium or high. So this tissue engineering model, it's like a big family of models. So there is like hundreds and hundreds of different models that are like developed for different application. And this model like now are really interesting for researcher because they can help to understand disease, because you have like a part of the complexity of an organism, but you can still like work easily with them. So in our lab, that is the McGuigan lab, so the PI is Alison McGuigan, it's what we're working on. So basically we're trying to build model that reflect the reality, and we use this model to understand like disease progression or like finding targets for diseases. So for like this talk, I'm gonna speak just about two models. There is other model, but I'm gonna speak about two models because I think it's the more like fun for you to know how it works. So I'm gonna speak about tracer, which is a model that is based on like, you know, like this cake, like the Swiss roll cake. It's basically that we sell. So that comes from there, the idea. And we use like cells that are layered in 3D structure. And you see that outside you have like the culture medium. So basically it's mimic what's the blood. So you have like all the oxygen, all the nutrient come from culture medium. And in the middle, you have like this aluminum mandrel where nothing goes through. So all that is like going in layer 3 or 4 or 5, so in the inner layer, go from outside. So you have this creation of gradient. And as you remember in the intro, I said that all these gradients are really important for like cell behavior. So this model is really kind of unique to look at graded environment. So I'm gonna focus on tracer, and after we will come back to the second model, which is glands. So how like we're doing this tracer model, how it look like basically. So to do tracer model, it's really simple. We use like a paper strip. So basically it's like tea bag paper. So you can kind of do it at home, right? You just like cut a strip of paper. After it's becoming more complicated because you have cancer cell on it. So don't do that at home. But like the scaffold is really, really easy. And we add this cell in a matrix in the paper, and that's make a strip with like cell everywhere on top. So you see that we have the cell with like a pipette. And after, like we take this strip and we roll it on an aluminum mandrel. So when you look at aluminum mandrel that is like cut from top, you see that all the region where there is cell are like stacked, and you have like this structure where it's like 3D layers. And if you think about it, where the culture medium is, bringing the oxygen and the nutrient. So this layer 1 have access to like a lot of oxygen, lot of nutrient, right? But this cell consume oxygen. So like when you go inner and inner, there is less oxygen, less nutrients. So you kind of like reproducing the fact that outside it's like a blood vessel, and on the middle is like the core of the tumor. So you created this gradient. And the big advantage is that model is that you can just unroll the strip. So you see like you cultivate the strip like that in your plate for like 2, 3, 4 days, basically as much as you want. And after you just unroll the strip, and as it's a strip where you know where you put the cell, you know after when you will make your analysis where the cell come from. So you can compare, for example, cells that were in layer 1 and cells that were in layer 6. And by doing that, it's really interesting because it's the same cell, but the cell was exposed to like high oxygen, high nutrient, and the other one was like low oxygen, low nutrient. So you can see how this gradient like influence cell behavior. And you can't really do that with a mouse, for example, because when you have the tumor out of the mouse, you don't know, like, let's say that you dissociate the tumor, so you end up like with a suspension of cell. You don't know which cell come from where, and that makes the analysis really complicated. So that's like a big, big advantage of this model, is that you keep the information, the spatial information of where the cell come from, and you can like correlate this information with how they look like. So their phenotype, how they respond to drug, how they proliferate, and everything. So this model, basically, it was done like 4 years ago, I think. And since that, we like improve it to use less cell, because like cell, you imagine that the first model, we were needing like millions and millions of cells. So we were spending like half of our time cultivating cell to just put them in paper. So it was really annoying, and there was not much time to do experiment. I mean, not much time. We were spending a lot of time cultivating the cell, but now with this new model, you see that all the blue area here, it's paper that is blocked with a polymer. So in this area, there is no cell. So basically using this model, we reduce the number of cells by 3 times. So it's much convenient for us. And especially if you think about like using patient cell at one point, you can't have like millions and millions of patient cells. It's like a precious sample. So by doing that, we open new possibility for the future. So the first step with this model was to characterize it, basically to show that this model was relevant to after like ask biological question. So as always, when you start with new thing, you have to start from the beginning, and you have to go like step by step. So as we're like thinking that that's a good model to create like gradient of oxygen, for example, first things to do, show that you have an oxygen gradient in your model, because if you don't have an oxygen gradient, you can't really claim that your model is good to create a gradient, right? So here what you see is that it's an immunostaining, so with antibody that reveal basically like cells that are hypoxic. And the more yellow orange it is, the more hypoxic are the cells. So the less oxygen they're exposed to. So if you look at layer 1, which is the layer on the outside, so the one that are close to the media or basically the blood vessel, you see that there is only blue. So blue is the nuclei, is just to show where the cells are. So you see that there is not much yellow. But as soon as you arrive as like layer 3, 4, 5, 6, you see that there is more and more cells that are hypoxic. So if you quantify this picture, you see that in layer 6 here, there is more like signals, so more like hypoxia than in layer 1, and you can keep this gradient for days. So that shows that the first claim that you're having like a graded environment work. After, you have to like show that basically it recapitulates what happened really in the tumor. So we took like few features that are like cell proliferation and cell death. So usually in a tumor, the cells that are outside of the tumor proliferate more than the cell and the core of the tumor. That is really simple. You have usually less oxygen, less nutrients, so it's harder for the cell to proliferate. It's like basically you have no food, no water, you will like grow less than people that have like water and food, right? So if you look at this graph, like EDU is the tool that we use to quantify the cell proliferation. And in layer 1, outside of the tumor or of the tracer, you have more cells that proliferate in layer 6. And that's correlated with cell death, meaning that it's logic. There is less cell that proliferate, less oxygen, so there is more cell that die in layer 6. And it's exactly what you see in tumor in humans, and you see that we recapitulate that. So with this model, we recapitulate like some feature of the tumor. But that is still really basic, right? Because you're adding only one cell type. And as I mentioned in the intro, like the tumor is like a bunch of different cell types. So since that, we start like moving like forward and thinking about like establishing co-culture model to look at, you can't go from like monoculture to the full model, right? You need to have like step betweens. So we are like 3 people in the lab that are working on different co-culture. And the goal is to know like, okay, fibroblasts and macrophage interact. So I'm working on fibroblasts and macrophage, interact this way. Ileana, which is a PhD student in the lab, is working on how macrophage and cancer cell interact. And Jose, which is another PhD student, is looking at how fibroblasts and cancer cell interact. And the goal is that, okay, we characterize all this co-culture, and maybe after we can make like a tri-culture. So with all these 3 cell type and look at the network more complex, because you need to like start from somewhere, right? So you can't just like jump into the complex model with 10 cell type again. I mean, you can, but it's too complicated, and the analysis after you will need what it's kind of control, right? You need to know how 2 cells interact, and after when you introduce the 3rd cell type, that tell you how this 3rd cell type modify. So I'm gonna speak more about like the fibroblast macrophage because it's my work, so it's easier for me. I know more like the subject, but like if you have any question about like cancer cell macrophage interaction or fibroblast cancer cell, you can email me and I will like transfer your question to Jose and Ileana, and I'm pretty sure that they will be super happy to answer because like they like outreach science and like speak about the research. So do not hesitate, or you can find them on Twitter or whatever. So why is it relevant to look at oxygen on fibroblast macrophage interaction? I mean, that's a good question. Why are we doing that, right? So in cancer, if you look at fibroblasts and macrophage, there are actually like 2 types of fibroblasts and a lot of type of macrophage. But if you take the big picture, there is macrophage on one side, fibroblasts that are activated on the other side, and fibroblasts that are non-activated, so the quiescent. And the role of this fibroblast is basically to like remodel the matrix. So they have like a huge role in metastasis, for example, because they can degrade the matrix. And when the matrix is degraded, the cancer cell can like move around and reach the blood circulation and go and to make metastasis, right? So these cells have a crucial role in cancer progression. And macrophage, on the other hand, have also a super important role because they're like implicated in like immune suppression. So I'm pretty sure that you heard in the news about like immunotherapy and like using immunology to treat cancer. That's like where macrophages are really important. And they're also implicated in metastasis and angiogenesis. And there is like already several studies that were done, but they were like not looking a lot about how the microenvironment influence this network. So for example, there is a bunch of study, so that's maybe like a little bit complicated, but it's just to show that all these cells communicate together by like cytokines, small molecule signaling. So they're always communicating and activating each other, regulating each other. And there is like really few studies that show that, for example, hypoxia can lead the normal fibroblast or the quiescent one into activate one that are like bad for cancer. And there is also some study that show that hypoxia can influence the behavior of macrophage. But nobody looked at the importance on hypoxia on the full network. And that's really important because that can reveal like new target for like cancer treatment, for example. That's why I'm working on this field. So how to answer to the big question, which is like how oxygen influence macrophage fibroblast interaction. So firstly, you need to create your model, right? So right now we're on a model where there is only cancer cell. So you need to make a good model where you have like the fibroblast and the macrophage run. After that, you need to like start deciphering the network. So basically how all these cells communicate and how that influence the drug response or like whatever you're interested in. And then when you know the network, you can identify and validate some targets that are relevant, for example, like for treatment, right? So I'm gonna go through like only the two first steps because the third step is not yet done and it's not the most interesting one because it's always the same approach. So when you want to establish a new co-culture model, it's exactly the same that for the monoculture model. You need to show that your model is relevant. So the first things that you need to show is that, okay, we're putting this cell in a paper strip. So that's kind of weird, right? You're introducing a new materials with the cell. So you have to show that this paper doesn't influence cell behavior by itself, because if it does, that's a problem because your cell don't behave normally when they're in paper, so you can't really make any conclusion. So to show that your cell like are not influenced by paper, you need to show that they're still like what we call plastic, meaning that they can still be activated or deactivated. So here, basically, like the more it's green, the more the cell is activated. And you see that when they spend time in the paper, they deactivate it, meaning that they still have this like possibility to deactivate or reactivate. But even when they are deactivated, if you put a molecule that you know activate them, they reactivate it. So these cells are still plastic. They can like still move around with their phenotype and respond to like extra cellular stimulus. So that's for the fibroblast. So that shows that they're still plastic. You need to do the same with macrophage. So that's a little bit complicated, but basically when you treat them with a molecule that is supposed to make them having like special feature, they have this special feature. When you treat them with another molecule, they have the other molecule special feature. So that means that they respond normally to what you expect. So that's basically show, okay, the paper doesn't influence the cell behavior. After, as you're mixing both cell type, you need also to like find the media, so culture media where both of them are happy, because if you have a media where one cell type die, you can't make a co-culture model. So to do that, there is like multiple ways to do it, but you can just say, okay, I'm gonna take fibroblasts and I'm gonna make them grow in macrophage media and fibroblast media. So the fibroblast media is where you know that they're happy, and I'm gonna see if they can survive in the macrophage media. And you see that the case, because like over time, there is the cell same number, so basically they proliferate at the same rate. So with all this like small experiment, it takes time to do, but it's like not really helpful to understand like the disease biology, but it's helpful to show that your model is relevant. And when you will ask questions using this model, you will be able to answer this question. And finally, as you're claiming that you want to look at oxygen, you have to show that you have oxygen gradient, exactly the same than before. Layer 1 here, no like red, orange signal here, a lot of orange. So there is an oxygen gradient. So we are happy with that. That means that the model is like kind of good to go. So that's like key bottleneck. We have still other confirmation to do before to be like 100% confident in the model. So like the next step will be, for example, okay, we know that happen in human when you mix, we know that this interaction between macrophage and fibroblasts happen in humans, and we will have to show that this interaction also happen in our model. And that's like the ultimate proof that your model is recapitulating what's happened. So when we will have all this data, we will be, okay, we are confident, now we can like try to decipher the network. So to decipher the network, we're gonna use like a bioinformatic approach. So I say like everything is future because due to COVID, basically we were stopped at the end of the characterization of the model, and all this like bioinformatics approach will be like in the coming months or year, depending on how stuff move forward. So using like single cell RNA technology, so it's a really new fancy technology that allows you to read all the RNA from one cell. So at the end, you have like all the protein from one cell, and you can do that on like thousands and thousands of cells. So when you have that, you have the information of cell A express this protein, this protein, this protein. Cell B express like protein A, B, and C. And you know, like, because we're working on with computational biology, so it's the lab of Gary Bader, and they created like kind of like a giant atlas of interaction that can happen. So they have like a database saying, oh, this protein can interact with this protein. And they have like that for like 115,000 interactions. So you can screen like a really big number of interaction. And using this like data and this library of interaction, you can calculate a score, and that tells you basically how likely the interaction occur. So you can know what interaction occur in layer 6, you can know what interaction occur in layer 1, and you can compare them. And by doing that, you can know the interaction that are modified by oxygen. And that's really cool, because if an interaction is modified by oxygen, you can be like, okay, there is less oxygen on the middle of the tumor. So if I target the interaction that are specific from the middle of the tumor, maybe that will be like really efficient as a targeted therapy, right? So when you like do this network, you have like thousands and thousands of possible interaction with scores. And after you just so that's like the computational approach. And after it's where you need biologists to be like, okay, this interaction, okay, it's modified by oxygen, but it doesn't matter for us because it's not answering a question. But this interaction that is implicated in drug resistance, we want to try that. And when you like identify all these targets, after you validate them and you see what it modify in tracer. So let's say that you show that interaction, I don't know, A and B is really important for drug resistance. You take your cell, you underexpress A and B, and you see how that influence like drug resistance. And if it influences, that show that A and B can be a potential therapeutic target for cancer treatment. So that's the next step. So again, after bioinformatics, so that will be more in years than months, but hopefully we will reach this point. So that's what I wanted to like told you about tracer. It's just to give you an overview of what kind of question you can ask with this model, how it works. And if you have like question at the end, we can discuss more about that. Now I'm gonna present like the second model, which is called GLANCE. And this model is not to look at graded environment, it's more to look at the region where two compartments, so where two cell type meet. So for example, as you remember in the tumor, you have like the fibroblast, you have the cancer cell, and there is like a border between these two cell type, right? And if you want to look at how cell behave at this specific location, GLANCE is the perfect model for that. So how GLANCE works? So basically it's used like microfabrication approach to make really, really tiny and really thin channel of hydrogel. And in this channel, you have two cell types. So you can have, let's say, cancer cell on the right and fibroblast on the left. So here you see a little bit more about how we did it. So you use like a plastic that you hot embass. So you make this tiny channel, and after you add like a slab of another type of really specific plastic. You seed the cell in a gel, the cell go into the channel, and after you kind of glue that under a well plate, and you end up with like a glance, which is the name of the model, and you can image that over time. So if you have a closer look at that, that means that you have like this model. So as you see here in green, it's cancer-associated fibroblast. In red, it's cancer cell. And you can look at how cell behave like here at this region where both cell type meet, or you can look at the behavior of cell here, like more far away, where basically they are not meeting the other cell type. And by doing that, for example, it's really interesting to look at cell invasion. So cell invasion is really important for metastasis. And the Elisa, like a previous PhD student, show, for example, that the cell at the interface region, the stromal cell form protrusion. You see that here, and it's not the case at the far from the interaction zone. So basically, when you have like cancer-associated fibroblasts and cancer cells that are really close, the cancer-associated fibroblasts change of shape, and that influences the way that cancer cell can go into the fibroblast compartment. And you can imagine that that means that it influences the way that cell can metastasis, right? So I'm not like working a lot on this model. That's why it was just to give like a really small example, but I'm gonna stop that on the model because also like time is flying really fast. So just no conclusion, but just like if you have to remember like few points about like this tissue engineering model, is that firstly, the perfect model doesn't exist. I mean, the perfect model exists. It's if you want to like study human biology, the perfect model is human, but like you realize that you can't do everything in humans, right? I mean, it's just like logic. So you have like a good model is a model that allows you to answer to your question. So if you're interested in like a question about gradient environment, choose a model for that, even if it's not really good for other like answering other questions. That doesn't mean that it's a bad model. It just means that it's not a model that is adapted to answer to this question. And that's like really, really important. Like if you continue into science, the choice of like the model of where you will do your research influence everything. So for example, if you're like choosing a different cell type, it can change totally the result. That's why it's always good to have like the big picture and think large and be, okay, I want to answer to this question, but even if it's not as good as other model in everything, if this one is like better in one field, I will choose this one. And finally, just like a really broad thought about like transdisciplinary research, it's really important. So it's quite recent, but you see that all this model, I'm a biologist, but all this model, it's not really biologist. Like it's like bioengineering, we're using like fabrication, so like scaffold, hot embossing, laser cutting, and all this approach, even like physics to look at the oxygen diffusion, and all of that is like physics, chemistry, biomaterials, and it's not biology. So like having like an interest in transdisciplinary approach allow you to open like new horizon. And I think it's really, really important now to don't just be focused on one field and be like, oh, I want to be like a immunologist only, but to be like, okay, I like immunology, but if something that comes from bioengineering can allow you to answer to your question, just use this tool. It's gonna be like much easier, and you will have like really interesting thinking and like science journey if you're like going into this field. So that's like for the science part. So I hope that you will have like a lot of questions about this model. And now like as the organizer request, I'm gonna like speak just a bit more about like how I end up like working in Toronto. So I come from here. So that's basically like the middle of nowhere in France. I've gone through all these places and I end up in Toronto, and I'm gonna just explain a little bit about my journey. So the European system is a bit different than the US system. I'm still not really familiar with the US system. I have like problem to understand it. But basically, in France, you do your Bachelor of Science, 3 years of research. I had the opportunity to make like a first research. My first research experience was during this Bachelor. So it was only like observation. I was just like looking at a researcher working. I had no authorization to touch anything, but that's already like a really cool opportunity. After the Bachelor of Science, I've done like a Master of Science in the University of Bordeaux, where I've done like 2 internships, one in like calcium signaling with a lot of like animal models. So I was like working on rat model. And after doing my second year of master, I've done an internship in like calcium signaling in cancer. And after that, I went through the PhD fellowship competition, because in France, there is a competition at the end of your master to have like basically money. And if you have money, you can do your PhD. If you don't have money, you're not allowed to do a PhD. And I've done my PhD, which is like 3 years in France, in University of Bordeaux. So I assume that Bordeaux, you only know it for the wine, but it's like a good university in the south of France. So basically, like on my PhD, when I start my PhD, my initial goal was, okay, we want to know if some calcium channel is like implicated in lymphoma dissemination. So lymphoma, it's a type of cancer. It's like what we call the liquid cancer. So it's like leukemia. It's a cancer like where the B cell are the cancer cell. And they're like circulating. So dissemination is the equivalent of metastasis, but we just use dissemination term for lymphoma. So I answer to this question, we make an article, but that was like really 2D biology, no bioengineering, no 3D model, no tissue engineering. So you can be like, oh, how I end up like working in this field. And it's kind of like really funny, because we were like, at the end, when we published this paper, we was like, okay, we have done that in 2D, but basically like in your body, the cells are in 3D, they're in blood vessel. So is this result are like kind of like transferable to what happened in vivo? And we were like, oh, let's see if we can like do that in a more relevant model. And exactly at the same time, there was like a talk at the university done by Dr. Pierre Nassois, which is like, is working with like bioengineering and like biophysics. And he introduced like the concept of like cellular capsule technology, which is a 3D model that is really cool. And we were like, oh, that's a good model. So maybe we can like try to put our cell in our model and see how it behaves and see if that can be a good model for dissemination. And it's basically how I end up working in 3D. And that was like 2 or 3 years after the start of my PhD. And we've done that. And actually like this model is really good. Like we reproduce what happened in lymphoma. And I've moved to this transdisciplinary approach, doing a lot of imaging, a lot of like biophysics. It was like the first time that I went to their lab, it was like, oh, what is that? There was so many like device and equipment that I never saw in a biology lab, but there was a biophysics lab. And it was like so cool to discover, like after 3 years doing kind of always the same, you just discover a whole new field with new toys. Basically you can like experiment so many stuff, and that's like sincerely like the best part of my PhD is when I moved to 3D, because everything was so exciting. So just what I want to say like by this kind of like journey is that it's not because like you're doing your first internship, because when we present research, like when I was in bachelor, they were kind of like, oh, if you start doing research in, I don't know, like immunology, you will be an immunologist like forever. And I think now it's kind of changing. And I think that's really cool, because that allows you to see like more stuff, have more fun, learn more techniques, see more people. So it's not because like you're working on one field on the beginning that you're gonna end up working on this field forever. And that's like not only my case, because I made like a small map to show you basically that's my journey is just like a normal journey, and kind of like in science, everybody has the same. So that's just like where all the people from my Gigan lab, so my lab come from. And you see that there is people from all around the world. And these people have background in computational biology, there is engineer, there is people that were from imaging, there is like people from all the background. And that's basically where we come from. But when you look at where all these people went for like just for science, to doing internship, to doing like research, you see that all of us have explored like so much place around the world. So mainly like Europe, US, Canada, but because also like there is a lot of people from Europe, so you kind of like stay close to your home. But like just if you have like one stuff to retain from like this like part, is that science is international. So do not hesitate to just like, if you have any opportunity to go abroad, even like if it's Ottawa or like another city in Canada or US, just go for it, because you will learn a lot of stuff. And if you have the opportunity to do interdisciplinary research, also I will like really encourage you to do so, because it's so fun to have like a skill palette that is like broad, and you can like understand, like maybe you're not as expert in one field, but you understand kind of like, how can I say, I lost my word. You can understand like the different field, and you can like try to integrate them, and that's like really, really cool, sincerely. So that's just like my vision of the research, but try and you will see, like, just take the opportunity. And that's all for me. Finally, I just want to, so that's like the McGuigan lab. So Alison, the PI, is there. I mentioned Rose, Ileana, and all of these people are PhD student or master student. So we are like a really young lab, and you see that it's really cool. Gary Bader Lab also, and Brendan, and also that's the financial support. So for example, like Medicine by Design is paying me for my PhD research. So that's like really important to highlight, that without these people, there would be like no research. And that's all for me. Yeah.
Thank you so much. That was really great. Yeah, so now we'll begin our Q&A period. So does any of the students who are a part of this have a question right off the bat? You can either type it in the chat box, or you can unmute yourself and ask Dr. Latour verbally. I think one question that I had that was just kind of came up based off of the tracer model. I was wondering how, because you mentioned that this was for looking at mostly like different conditions that might affect cells. So I was wondering, have you just looked at, it kind of looked like you had done at least 6 layers, or like how many layers exactly like have you guys tested with the different conditions?
So I will say like it depends, right? So it depends on the cell type. So for example, if you want to create an oxygen gradient, you just need cell that consume the oxygen. So if the cell that are on the layer 1 consume a lot of oxygen, you will already have hypoxia in layer 2. But if your cell like consume less oxygen, you will need more layers. And also it depends if you want to go like to really, really hypoxic environment, or just to an environment where you have a little bit of hypoxia. So by playing on the density of cell or on the number of layers, you can like modify all these features.
Okay, cool, thank you. There's another question I had was, so based off the glance model, and this might just be because I'm not as familiar with the cancer research and the past history with it. But because you said you can kind of compare the competitiveness between 2 different cell types. So I was wondering if maybe you guys have already done this just with initial testing, but have you looked at like healthy cells versus unhealthy cells, and the differences between them, and if like unhealthy ones, like how fast they're able to outcompete the healthy cells?
You mean like LC, like normal cell versus
Yeah, like
cancer cell?
non-cancerous, yeah.
No, we never tried that. But basically, like you can like likely do that. Put cancer cell on one side, LC cell on the other side. I mean, when you put LC cell, the big question is which kind of LC cell you will put, right? Because it can be LC fibroblasts even. It can be like endothelial cell to mimic like the blood vessel. It can be a lot of stuff. But the big advantage of this model is that this hydrogel layer is really thin. So you can like image on the plate, and it's high throughput. So for example, you can like put different compounds, different drugs in your wells, and just by imaging, you can have an idea of what reduced, like for example, the invasion of the cancer cell in the non-cancer cell.
That's so cool. Looks like we have a question in the chat. So are your models specific for any certain types of cancers, or can this be widely applied to like all types basically?
So Actually, like it's not for one type of cancer, and it's even more, it's not only for cancer. So we have people that are like working on obesity. And when you have obesity, there is like a decrease in oxygen, for example, and there is people that are putting like fat cells in that, and that are reproducing like the obese tissue environment. And even like for the fibroblast-macrophage interaction that I'm working on, these two cell types are really important in other contexts. So it's not only on cancer, but for example, when you have like wound, like you disrupt the blood vessel, right? So you have no oxygen supply, and that makes this environment really close and to a fibrotic environment or cancer environment, because you have like fibroblasts that come, you have like macrophages that come to the, and there is less oxygen. So this type of question, you can use after your data to look at fibrosis, for example, or wound. It's not even only for cancer.
Next question is, so as you guys continually make progress on your model, how similar do you think you can get it to being to an in vivo system?
Oh, that's a really hard question. I mean, it's a really good question, but the really big problem, I think, is the vascularization. Because, okay, we're mimicking like oxygen gradient, but when you have a tumor, you have some blood vessel that goes through the tumor. So it's not only like radial, right? So I think a really, really big question in the field of like tissue engineering in general is how to make vascularization and how to make blood vessel. And after you need to integrate that in the full model. And another big problem, like challenge, I will say challenge, because that will maybe like happen at one point, is to find like the media, for example, that fit to all the cells. Because like right now we have like only 2 cell types. So I'm quite lucky because I have my media, the normal media that is like working for both. But for example, there is co-culture where it's more complicated and you need to create a new media by taking a basic media, adding some like elements that all the cells are happy. So imagine that if you want to reproduce a real tumor with all these components, you will have like, I don't know, 20 cell type. So you have to find the media that fit to 20 cell type, and that can be only finding the media can needs like years of research.
Yeah, gets really complicated. Okay, I think maybe this will be the last question as we have another talk coming up really quickly. So somebody would just like to know if you can go a little bit more in depth how using bioinformatics helps you to basically find your ideal targets.
So the assumption is that if you have a cell that expresses A, the other cell that expresses B, and you know that A and B can interact, if there is more expression of A and more expression on B in one condition than in the other, you know that it's more likely that this interaction is happening, right? So by looking at the level of expression in the different cell, because again, with the single cell RNA technology, you have all the expression level of all protein in one cell, and you have that for all the cells. So you have that for all the macrophage, in our case, you have that for all the fibroblasts. And you can like look at the mean expression by the cell of expression of ligand A, for example, expression of ligand B, and compare that in layer 1 and 6. And you can like, basically it's when I say it's a score, it's just give you a level of confidence that this interaction is happening. So bioinformatics will not tell you like this is happening for sure. It tells you like, oh, maybe that happened, and it's happened more likely in condition A than in condition B. But after, like, you definitely have to validate this finding. You can't, for example, be like, oh, we revealed this target with bioinformatics, let's just give up to people a drug that is targeting that. You need like a full step of validation. And what is like even more fun or more exciting is to, after you validate, to understand how it works, because like it's already really important to know the mechanism of action of like molecular interaction. So you can like, I think like Gary Bader Lab have a website with a kind of like description of this concept in like really non-expert language. That was really helpful for me because like I've never done bioinformatics before. So, and you can even like contact me or them and we can like discuss later if we don't have much time. But yeah, you just like infer what is possibly happening from the expression level in both cell type.
Okay, great, thank you. So thank you again, Dr. Latour, for this wonderful talk and for sticking around to answer a few questions from the students. Yeah, if anybody would like to get into contact with him, I shared a research talk webinar, and in there has 2 emails for our team lead and our internship, the director for this, and we can put you into contact with him. That way, if you're interested in learning more about this.